Welcome back. I hope everybody had a wonderful lunch. I know we had to scarf some food down here, but we're back and ready to go. So our second concept presentation today is a tag team from Liam O'Fallon from our population health branch and Adam Berger from the NIH Office of Scientific Policy, OSP. So this is going to be a continuing conversation that we actually started, I think, 3 councils ago on the return of results for non-genetic data. So Liam, if you could run us through your concept. Thank you.
Sounds great. Thank you very much, David, for that. And good afternoon, everybody. As David said, my name is Liam O'Fallon, and it is my pleasure to be presenting to you this concept on, as the title says, well, let me start over again. Sorry, good afternoon. It's a pleasure to present to you the following concept that we have developed in partnership with the NIH Office of Science Policy and the All of Us program, and most recently with the National Human Genome Research Institute, or NHGRI. This has been an effort that has evolved and matured since our first council discussion last February, as David said. Over the next 20 minutes, I will review the context, describe the general concept, and outline the rationale for it. At the end, I look forward to addressing the questions and listening to feedback you may have. So report back has been referred to as a team sport and, because of the many disciplines that are needed. Here are my teammates who have contributed extensively to the FOA concept, and I have enjoyed working with them. I'm pleased that Adam Berger, as David mentioned, from the Office of Science Policy is joining us remotely. He's going to be making a few comments at the end of my presentation and will be participating in the Q&A session. I'd like to ground our conversation with some quick definitions. RBRR is an acronym that I will be using throughout the presentation today. It stands for Report Back of Research Results, and you can see on this slide how we are defining it. For the purposes of the concept, I will be referring to reporting back individual data in the context of environmental health, non-genomic, and gene environment, or G/E research. And you can see the definitions here on the screen, which have all been emerging conversations in the context of report back. Yesterday, I thought that Dr. Wychik set the stage nicely for my presentation and the value of reporting back research results. In his presentation, he emphasized the importance of community engagement in the context of environmental justice, health equity, and climate change. In addition, he, and earlier today, Dr. Shui, noted the emerging work in exposome research that included the use of community-level exposomics to address health disparities and inequities. Within the extramural research community, there has been a clear message about the importance of report back, which I've listed here. Report back of research results is particularly important to community-engaged research projects and any project in which participants are providing their data to answer research questions. By returning results to participants, researchers are building trust with their participants or their community partners. They are demonstrating respect to the participants. After all, they are contributing their time and data. It increases the transparency about the research process and findings. It fosters increased engagement in research as people learn about the findings. It strengthens both science literacy and environmental health literacy of the participants as they see and understand their results. And it increases or empowers the community members. Additionally, participants of a recent gene environment meeting noted that research shows that nearly everyone wants to know their chemical exposure or their G/E results, even if the health implications are uncertain. However, please note that while there is a consensus that reporting back research results is important and that most people want to receive their data, we acknowledge that some individuals will not want to receive their data and as such have the right to opt out. So if there are so many benefits and many participants wish to receive their data, why am I up here virtually? So while there are numerous anecdotal benefits, there remain many questions and unknowns about reporting back research results. So I'm here today because our collaborative team posits that this concept will help to advance the science of reporting back environmental health research non-genomic research, and G/E results. This program will provide us with a better understanding of different perspectives of RBRR and how environmental health and non-genomic data can be returned to participants and communities effectively and properly. As conceived, this program will help to establish new tools and approaches to report back, as well as adapt, implement, and test existing tools and approaches from different settings. We believe that the program is positioned to highlight the necessary supports, training, and guidance to ensure feasible, appropriate, and quality report back. It's hoped that the program will enable consistency of report back across institutions and provide institutional review boards, or IRBs, with the necessary information to help with their own decision-making. Given the focus and the desired outcomes of this concept, we see it as an opportunity that will require multidisciplinary investigator teams that are already doing work in report back or are preparing to report back environmental health, non-genomic, or G/E research data. This concept aligns closely with the following themes and goals of the current NAEHS strategic plan. And don't forget that you can add your feedback for the new 2024-2028 NAEHS strategic plan as well. So if you want to provide feedback on report back, please weigh in and provide it there. We are bringing this concept to council now based on the outcomes and recommendations from the following 3 events shown in the green box that took place focused on report back of research results. The first was the National Academy of Sciences, Engineering, and Medicine, which released a hefty 399-page report in 2018. The report was commissioned by the Centers for Medicare and Medicaid Services, the Food and Drug Administration, and the NIH. In the same year, the Partnerships for Environmental Public Health grantee convened a grantee meeting focused on the same topic of reporting back research results in the context of environmental public health. The G/E Ethical, Legal, Social Implications meeting was co-hosted by NIEHS and NHGRI, produced a few recommendations. And I'm showing here sort of a combination of the various recommendations that came out of those 3 events. First, the acknowledged need for dedicated guidance, expertise, and resources to support high-quality report back. There's a need for guidelines and expectations to be clarified. Training and capacity building are required for quality report back. Resources for implementing it are vital. And finally, research is needed to improve report back methods, to focus on best practices for giving participants access to resources to do something about the data that's provided, on best practices for focusing report back on actionability, and for how people are using the report back to modify their behavior. In addition, last February, as David said, we started a council conversation on reporting back environmental health research results. The goals of that first session are shown here on the screen on the left-hand side. We heard from Dr. Katrina Korfmacher and Dr. Julia Brody, who talked about the context, and then we talked about the context and the need for report back, as well as the state of the science. After those discussions, we were encouraged to develop some recommendations for the National Institute of Environmental Health Sciences to follow up on. In June of 2022, I came back to council and presented a series of recommendations outlined in the document moving forward with reporting back environmental health research results. I developed that in partnership with Drs. Korfmacher and Brody. In that presentation, I noted the importance of coordinating with the NIH Office of Science Policy because of their expressed interest in the bioethics of reporting back research results. At the end of the council discussion, there was general support for moving forward to advance these recommendations. Shortly after the June Council, Adam Berger, who will be on the call today, from the division, who is the division director of the Clinical and Healthcare Research Policy at OSP, reached out to say that his office would like to co-develop a research program focused on report back of environmental health and other non-genomic research results. He noted that there was plenty of research in genomics, but that there was a need in the environmental health and non-genomic space. Dr. Wojcik then put us in contact with the All of Us program because they were discussing how to incorporate report back into their work. So in the context of these recent events and the ongoing discussions, I'm bringing to council the concept to support multidisciplinary research focused on reporting back research results grounded in a bioethics framework. As noted, the research enterprise is increasingly moving towards greater return of individual specific research results to increase the value for participants and to improve the scientific rigor and reproducibility. While report back has been commonly discussed in relation to the return of genomic research results, there's been a growing movement towards greater report back of environmental health, non-genomic, and gene by environment research results. That said, there are recognized challenges in returning those results. As has been discussed in these meetings, there's a need for research to inform community engagement approaches, guidelines, and additional educational resources for effective communication of research results to individual participants, and as appropriate, aggregate results to other key partners, including community-based organizations, tribal nations, public health departments, healthcare professionals, policymakers, and institutional review boards. Based on the literature and the recommendations from the 3 meetings I mentioned, this proposed concept would encourage multidisciplinary research that would address at least 2 of the following 3 topics: health equity, communications approaches, and data use. Since this concept is grounded in a bioethics framework, all proposals will require a clearly articulated bioethics issue, such as, but not limited to, data sharing, participant privacy, potential stigma, and/or discrimination. And while not required, the use of community-engaged research approaches will be highly encouraged. Health equity is a topic of great importance in the context and focus of community-engaged research, environmental health disparities, and environmental justice. It's a timely topic with regard to climate change and the executive order that focuses in on health equity and climate change in health. As we heard yesterday, it could also be very helpful in the emerging area of exposomics research. Furthermore, community groups that face disproportionate exposures or disproportionate health outcomes have been at the forefront of report back. They have expressed a desire to know what they are exposed to and to use it to inform public health action. Such projects can examine the role of report back in reducing and/or mitigating the impacts of health disparities. Projects could examine the additional resources, tools, and trainings that are needed for individuals, communities to use report back information to address these disproportionate environmental exposures. We are aware of different report back approaches and that have been implemented successfully in one community or population, but it's not clear if that approach can work in other places. As such, research projects evaluating the scalability and sustainability across multiple interested parties and groups would also be encouraged. Equity also includes issues such as access to information. Therefore, projects could examine the equitable implementation of report back. For example, strategies to ensure that report back reaches all populations in a way that is tailored to their individual needs, language, etc. Communication approaches. We are aware also of digital communication tools and print materials for reporting back data. We're keenly aware that genomics field already has tools that could be useful to environmental health and non-genomic report back. However, we need to understand how these tools and approaches could benefit both the research participant or community member, as well as the scientific question. So what is needed to successfully increase environmental health literacy or science literacy, as well as to address issues of language justice? It is essential to build capacities of researchers and others to do report back effectively. As such, research could include examining methods and tools that make reporting back research results more accessible and translatable to research participants or other interested parties or partners. Could include applications proposing to test the feasibility and acceptability of adapting tools for reporting back other results, such as genetic data, to environmental health results, as well as applications assessing risks or benefits of using emerging approaches, such as electronic report back. Using the data reported back, We assume that people are going to use the information that they receive. However, we'd like to better understand if they are acting on it and or who they are taking it to. Research could focus on how people respond to and or use report back information, which could include behavioral processes involved in a participant's understanding and acting on those results. We know that participants often share their results with their family healthcare provider. We just heard a good conversation in the last session about the preparation of clinicians. Therefore, it's going to be important to build the capacity of clinicians to enable them to communicate or translate results to patients. Testing and evaluating new or adapting training approaches for clinicians could be a part of this theme. So why is this concept important and needed now? This concept will provide us with the scientific foundation for informing and developing guidelines, training materials, and other resources to ensure quality report back. In other words, this program will help to meet the stated recommendations from the events mentioned earlier. It will address knowledge gaps. Reporting back of environmental health and non-genomic data is taking place, but there is a need to test and examine these approaches. The use of these findings will help to inform the guidelines for future work. It's relevant to current conversations, climate change, environmental justice, disaster response, and data management, as well as DEIA. All of these areas benefit from quality report back because work depends on building and sustaining trust. Finally, it addresses the needs for sustained funding. Bioethics supplements have been generating interest in this area of work. This concept would advance the science in our current understanding by enabling multidisciplinary teams to examine thoughtfully the report back of environmental health, non-genomic, and G/E data. So now I'd like to turn the virtual podium over to Adam Berger from the Office of Science Policy at NIH. Adam, thanks for being with us today. The floor is yours.
Thanks, Liam. A presentation. Very much appreciate all of your and the entire team's efforts to really bring this to fruition. I'd also like to thank Council and Dr. Wojcik for your interest and enthusiasm over multiple meetings for this topic. As Liam mentioned, my name is Adam Berger, and I'm the Director of the Division of Clinical and Healthcare Research Policy in the Office of Science Policy. It's actually part of the Director's Office here at NIH. We are really thrilled to be collaborating with you, the All of Us Research Program, and NHGRI in this concept. Partnerships like this are a major component of our larger bioethics program itself. In our view, bioethics is foundational to the entirety of our policy work, which really seeks to anticipate and plan for potential ethical implications for the research that NIH supports. Integrating bioethics into biomedical and behavioral research can really enhance the translation of research into practice, and it ensures we are engaging with research participants and communities in an inclusive and appropriate manner. It also makes sure that we are considering the outputs of the research that will be used and the findings that will be taken up. Simply put, in our view, taking bioethics into consideration just leads to better science. Report back is one of those quintessential bioethics issues. This concept and the studies that are funded under it will help us better understand the to whom, what, and how of returning research data to participants. And it will help develop a robust bioethics research evidentiary base to inform policy as we are developing it. I do want to give major kudos to all of you for taking a leadership role, both in recognizing the value that individual level research data has to participants and the scientific enterprise, and also for seeking out best practices to enable appropriate report back. As Liam mentioned, there are a number of significant benefits to reporting back research results. It ensures that research participants are being respected as equal partners in research. This is a major impetus for us as an agency over the last several years. It also increases transparency, it builds trust, and importantly, it fosters engagement in research. This concept is going to help expand on and leverage that robust evidentiary base that Liam mentioned earlier that already exists for return of genomic research results. And our hope is that this will actually be able to inform the return of environmental and non-genomic results. Building on that evidentiary base, we think it can provide NIH and the research community with data on different perspectives and how environmental and non-genomic research data can be returned to participants and communities, both appropriately and effectively. Research funded through this concept may also result in the development or testing of tools to advance capabilities to return results in different settings and to different populations. ultimately at the end of the day, we want to make sure that we have robust engagement throughout the country and throughout all different populations that reside here. We hope the projects funded through this concept will end up empowering researchers to return results in an ethical, timely, and appropriate way, and enable NIH to help build a facilitating environment to return. As noted earlier, we are extremely excited about this partnership, and we look forward to seeing the creative approaches that the community comes up with to address report back. Liam, at this point, I'm going to turn it back to you to kick off the Q&A.
Thank you very much, Adam, for the comments there. And just in closing, again, the purpose is to support multidisciplinary research projects, identify, develop, and adapt test strategies for reporting back environmental health, non-genomic data to study participants and key partners. And Adam succinctly tied up the outcomes that would come out of this. So at this point, thank you all very much for your time and attention. Now I'd like to invite comments from our two council reviewers, Drs. Ingram and Goldman. Dr. Ingram?
Great, thank you, Liam. And so we had a pretty robust discussion amongst the three of us about this concept. And so just some of the things that came out of that discussion was just thinking about the difference between reporting back when you have more like human biospecimens. So those have been overseen by an institutional review board as opposed to true environmental samples, say air sampling or water sampling, different types of things like that, that don't have that same type of oversight. And so one of the issues in my mind, just because I've seen this in mostly tribal communities, is people are attempting to disseminate or provide that information back, but sometimes the communities didn't even know they collected those environmental samples. And so it's just really important for us thinking about reporting back, but also, what is the timeline, the process from start to finish, so that folks, will be able to say, I knew this group was doing this work in our community, and I expect to see what the results are, and then be able to get those back. Whereas with the biospecimens, I feel like that is very much has a lot more oversight. So it's just something that I think researchers need to be sure of. And then I think I'm really excited about the opportunity to look at different ways of reporting back to different types of communities, because again, I feel like sometimes as a researcher, it is a lot of work to make sure that those communities get your results. And sometimes it's easy just to say, well, we posted flyers, we did this or that, check the box and we're done. So I guess I'd strongly comment that there would need to be an assessment as to whether it worked or not, the report back. I know in our own work, we have tried different ways to report results back to tribal communities, and some of it is just based on knowing where they get their communication. So that community engagement seems very important. I guess it's not going to be required, but if you don't have the community telling you, people aren't looking at their emails, they listen to this station on the radio or whatever the case might be. It's super important information. So, and then I guess I'm trying to think, I had one more point, but now I've lost it. So I'll just hand it over to Lynn. I can't remember. If I remember it, I'll come back on.
And you're on mute.
I was thanking you and welcoming you to chime in at any time, Janice. I think we're very much in agreement. I think this is a great initiative. I think, Adam, it's important to understand people in environmental health have been very dedicated to trying to do these report backs over a very long time. And this has to do with our concerns about environmental justice, our concerns about the fact that most of our research, much of it is community-based and participatory, and we need the trust of the people we work with, but also our commitment to something called right to know, which is a broader ethical concept, even than what IRBs look at. And sometimes, and I think why we need the research, sometimes our IRBs can be the biggest obstacle to being able to do this, because they rightfully say, okay, are we doing more harm than good if we're reporting information that we don't have a a precise context around what it actually means to the individual person's health. What is the benefit to that person of getting that information? It's the kind of question that I've heard. That is, when you're dealing with a CLIA lab test, it's a clinical test, and they can take that to their doctor, and it can be interpreted in the context of their own health. It's different than when they're dealing with, say, a biomonitoring test, where the doctors don't understand what that means, or even worse, how do they interpret? They don't know how to interpret the test of your tap water, your air, your soil, your dust. And so that is, I think, something that we all believe needs to be communicated, but there's a lot of skepticism about that. And I think for good reason and that's some of the reasons that the research is needed. We already heard earlier today in the Exposome discussion about the fact that, round robin show that a lot of these environmental labs don't actually produce consistent results. They can tell you if your soil generally has a lower or higher level than someone else's soil of something. But if I'm comparing an exact number from a lab in Denmark with a lab here and a lab in California, they're not necessarily consistent. And so that's an issue, and it needs to be understood by people that that some of these measurements don't have exact yardsticks yet. Some are really great, like lead, where there are all these proficiency testing programs, but a lot of them aren't that great. And so that just is another thing that adds to the complexity around context. It's also true, and in my own experience, the units that we use to measure things are not generally understood by the general public. So I've had people tell me that, parts per billion, that sounds like a lot more than a part per million, right? Where it's just absolutely the converse. But members of the public aren't often comfortable thinking in terms of ratios and the ways that we communicate and learning. And I think research can help with that, learning how to communicate these findings in a way that help people to understand the magnitude and the relative magnitudes of numbers. So, that point, I think that, Liam, you made in your slide, that data to knowledge is the key, that it's communicating the knowledge that's important and how to do that. But I also think that we have to consider the capacity of communities to be able to absorb the information. And I hope some of the research might have to do with the ability of researchers to help with building that capacity. Is it possible, when presenting results, to provide information that might be helpful to healthcare providers who need to then interpret it for people in the community or other people in the community who can serve as experts? Janice mentioned the issue of, doing, community-based research, but researchers also can get involved with bringing in expertise to help to advise community members. And I think with some of the work that we do, that can be really, really important, that we're building some capacity in the community to be able to understand and interpret these results, so that the people in the community aren't just left there with a sea of numbers that just may seem scary, but isn't actionable or isn't helping them to identify what they should be doing, to prioritize actions that protect their health or the health of their families. So I think you're hearing a very, very strong support from us for this, that it is a daunting task. It's even more complicated, I think, than genomics. I think it's even more complicated because, at least with genomics, physicians, nurses, healthcare providers have some general framework for understanding that. But most healthcare providers receive more than just a microscopic amount of education in environmental health. And you just cannot assume that there are people in the community who are going to be confident about being able to use this knowledge to be able to take action. So anyway, but thanks very much. I think it's an important initiative, and I hope that to see the findings at some point. I wanted to make one other point too, and which wasn't raised, but we are now at this juncture where all of our research findings are going into publicly accessible data sets. And these publicly accessible data sets, many of them are likely to be accessed by data scientists and other people who are not experts in environmental health or epidemiology or exposure science or any of the other science areas. And they may be generating findings from data science type explorations of these data. And what will be the obligations that those secondary users of these data will have for communicating those findings back to the people from whom those data were collected. I think that that's maybe not an issue for this particular exercise, Liam. But I think that this is going to be more and more a reality, that people are going to be obtaining information, not just from the scientists who worked with them, maybe did the informed consent process with them, maybe were out there in the community collecting the data, but other scientists who are doing these secondary analyses and who don't have any relationship to the members of the community.
Sounds like a good bioethics kind of question, right?
Liam, I just wanted to add on the action. So just,
Yeah.
and I don't know if it's this mechanism but in the dissemination or reporting back, what is the person who gets that information, what are they supposed to do about it? And so there could be just on their part, I need to change my health habits or whatever. But I know in a lot of the communities we work in, there needs to be something done by another entity like the government to clean up or something. And you have to be prepared to really talk about that as well, because I've heard communities say, you all have researched us to death. When is this going to get fixed? So just thinking about those next steps, and it may not necessarily be your research lab, but just it makes the dissemination of the report back very uncomfortable. If you just tell them you have arsenic in your urine, you have these problems, have a nice day. So just trying to think about that's something in these, these conversations that you have, just where do things go? So just keeping that in mind, because sometimes I know I've talked to people that know there isn't an answer and they're very concerned. Kind of what Lynn was saying too about, if there's not an actionable thing, do you really want to give them the information? Well, that's to me ethically wrong, but just thinking about the next steps.
Oh, I want to just tag on to that. I could tell a million stories, but I won't do it. But I do think that this is a major issue. Researchers generate data that do prompt action, but they don't necessarily know what that action might be, or they're not necessarily comfortable providing public health advice to communities. And I personally think it's important that they are in communication with the people who do local public and environmental health, and that those individuals have access to the data in a timely fashion to be able to analyze them and provide those results. I used to work in state public health in California, and all of my stories are around researchers at the last minute dumping out data. Okay, so what are we going to do about the fish? What are we going to do about the drinking water? When it could have been, there could have been that coordination, and that creates a lot of anxiety. And then you get back to why the IRBs, are worried, that you could do more harm than good. So.
Thank you both. So David, if it's all right, should we just go ahead and open it up? I see Gary's got his hand up.
Yep, Gary would be first in the queue.
Yeah, so Lynn points out that this may be harder than we have for genetics. And I want to point out that there's a National Society of Genetic Counselors with master's degrees all over the place. Like there's a whole field on the easier genetic side. And we have to think about how you build up the professional infrastructure to provide this, because it is more complicated. And if we're sitting here with no organization, no professional organization, it's going to be really difficult to make this work.
Yeah, just to second that was one of the major things that came up actually in our Exposed Home series last summer was, returning this data has to be done in a way that is interpretable. And we need a field of people that is their expertise, is helping to interpret this data. So very much agree with that, Gary. Trevor, you're next in the queue.
Liam, this is a great presentation, and we certainly need guidance in this area. I was wondering if any thought had been put into the point that was made about doing more harm than good, in the sense that, let's say that you're living in a community and the soil is contaminated, and there's also been biomonitoring and biomarker work to indicate individuals actually have levels in their blood or urine that, you know, are of concern. If that information is then made public, what does that do for home values? What does it do for health insurance of those individuals?
So Trevor, I think that those are, you know, clearly some of the conversations that we would seek to get at in the context of the ethical implications of reporting back research results and in determining that. So, you know, hopefully there would be some of those conversations to help us out as we move down the road. What are the guidances? How, you know, what are some of those determining factors? Adam, anything else to add?
No, I think it's a great point, and one that certainly needs to be taken into account. You know, if I can make an analogy to the genetics and genomics field, it is very similar and reminiscent to what concerns were about making genetic information known, but from a different standpoint. We weren't talking about it from home values, but rather, you know, health insurance or discrimination at the job, you know, at your job site. And so it helps to make sure that we understand what the implications are, and that if there's a potential mitigation step that can be put in, that those are made available. So I certainly hope that some of the research that, you know, research concepts that are put in for this will help to address some of what everyone has been talking about, because I agree completely. The infrastructure and supports necessary to be able to return. There isn't a genetic counselor equivalent that I'm aware of in the environmental health space. So, I mean, those are things that, you know, we need to be cognizant of to make sure that we're understanding what's needed in order to facilitate this. So, you know, again, I think this is going to be looking to the research community to help facilitate identification of gaps, understand what the landscape looks like, and really bring potential tools to help resolve this.
The other comment I had was that in terms of reporting back research results, you know, the community that's been exposed is only one stakeholder. At what point does the polluter get told? And not every polluter is a bad actor. You know, some industry want to do the right thing here, and so they might appreciate getting a heads up about possible emissions or contaminants that inadvertently have arisen.
Andrew Giller, I think you're next in the queue.
Thank you. And Liam, I'm very, very excited to see that you're embarking on this research. For us at EPA, I think we face some of the issues again that Trevor raised, both at the local level and at the national level. So thinking about report back, just in terms of releasing things like national maps of lead exposure or lead exposure potential, and the goals, for example, of a lead poisoning prevention program to elicit more higher compliance with surveillance versus those other unintended consequences, like I can't sell my house anymore because you put it in a high lead exposure area. These are the kinds of risk communication issues that we wrestle with as we start developing national pictures of potential lead exposure, potential PFAS exposure, other toxicants. So I hope that as you move forward, these are the kinds of issues that that you're able to develop approaches to. A quick question. Rick summarized some of the community engagement efforts that that NAEHS has just embarked on. Have these kinds of issues come up in the early discussions that you've had with with your community partners?
In the context of the unintended consequences or the importance of reporting back research results?
Probably the latter, the importance of reporting back.
Obviously, this is a hot topic within the research to action and our partnerships for environmental public health, you know, in the context of community partners wanting to hear what their results are and what they're exposed to and how they can take action. So this is something that is being talked about quite actively.
Thanks.
Harry, you're next.
Thanks, Liam. It was a really interesting topic, and I think worthy of consideration for funding. One of the things that I'm wondering about is, you know, how nowadays most people, or a lot of people, get their information disseminated, actually misinformation and disinformation disseminated on social media, and whether or not there isn't a role here for research into the effects of social media on the quality of data, its interpretation, and so forth, and its dissemination. And in that regard, do you think it would be interesting to include some of the companies like Meta and others that are in charge of in some ways a gateway for social media to also have their extensive research backgrounds engaged in some of this work?
A really interesting concept, Terry. Thank you very much for that that idea.
Then you're next in the queue.
Thank you. Yeah, I thought that was an apt point about real estate values. And I think that many of us who've done a lot of work in communities realize that the realtors, banks, industry, they're part of the community. The community is not just residents, it's the whole fabric that holds them. And I think you err when you don't take that into account when you're going out there to work with the community. But it's not just the value of the property, and this may be more like what you see, Adam, with the genetics research. There is a concern about stigma that can result from the knowledge about some of these things. One piece of research I was involved with a long time ago with an American Indian community with mercury, where we spent a lot of time, many months, actually talking to the community before we released results, because they felt their children were already stigmatized at the schools and are already, because of the color of their skin, they were treated as though they didn't have as much academic potential, and they didn't want, therefore, the state health department talking about mercury exposures to their kids, which would have reinforced that stigma. And I think that happens with genetics as well. And it's something that is an enormous issue with some of these environmental findings. So anyway.
Perfect.
And Lynn, that is one of the called out areas as an example only, but one of the called out areas that we've incorporated into the FOA itself, or into the concept at this point, specifically because we want to make sure that those issues are addressed by the community.
Yeah, hi. Can you hear me?
Yep.
Oh,
Go
yeah.
ahead, Irva. Thank
Okay.
you.
Go ahead. Yeah. So a few comments and thoughts. Well, first of all, I want to thank you, Johnny, for commenting about all the things we measure that we don't really know how to interpret, and that giving that kind of information back would be frowned upon. And I recall now we had a community advisory group on when we were first starting our work on autism, and as we were, they were asking about the design and what we're planning to measure. And they said, and we are going to get all of that back, aren't we? You are going to give all of that back. And we said, well, no, we will give back what is meaningful, what would, things like lead levels or mercury, or things where we have some standard, we know what the harms are, and we also know what can be done. And we never convinced them, but we sort of agreed to disagree. And, I think it was good that it was aired, that there were these opinions that were differed from what we felt would be good. And the other part that we were really worried about was that not only were there many things we were measuring that we didn't know much about, or the scientific field doesn't know much about, but that it would take a huge amount of effort to even become expert, at all of the different chemicals we were planning to look at, to be able to, do a good job of counseling people in some way. The other, a few other points, I think that one of the positive things about reporting back is that if done well, it sort of stimulates interest in environmental health and in science. And we live in a country where science, there's a strong anti-science element out there. And so I think having this happen on a broad scale across studies all across the country could really have some impact in terms of getting people a lot more interested and curious and respectful of science and environmental health science in particular. The question of the stigma associated with communities is actually really different from the question of the report back. As I understood it and saw from your slides, it's very much, this is the individual information being passed on to an individual who can do with it what they wish. Which is different from when we publish a paper with a map identifying regions. Maybe some of the issues are similar and related, but I think the privacy aspect of this is really important. And that's really separate from, what is appropriate to publish in terms of, it isn't just about that it could identify specific individuals, but that, as Lynn pointed out, that an entire, region ends up with the stigma or even just the worry or guilt or, many emotions that come up when people learn about some of the harmful things they may be exposed to or may have had behaviors that personally unintentionally may have harmed, their families or people around them. Another thing I wanted to say is that working with Native American populations, that there's a lot of, history around many of these populations when they have participated in studies and that they control and have access to all of the data that we produce and control what happens to those data. And they have, at least tribes that I've worked with or am aware of, or students of mine have worked with. There's, many of the tribes have strong environmental departments within their nation. And some of them aren't recognized as nations, but they also have those, including people who are very well educated on environmental health and environment, and they know their environment intimately from, really generations of experience. So I think there may be some models that investigators can look at in deciding how they're going to communicate back to their, the form of what it's going to look like when they present the data. And then lastly, I may have, I don't know if I dreamed this or what, but did I see some RFAs out already with saying that had requirements for a plan for report back and some formula for the funding that can go towards that? Or did I see that in an RFA or?
There might be and Adam, you can probably address this a little bit more, but there might be a supplement that you had seen out there, bioethics supplement.
Okay. Yeah, I mean, I think that has been something that has been intimidating to a lot of researchers writing grants is we don't have the funding to do this. And so I think that is a really critical component that it's, cramming a lot of things into these grants that have maximum that haven't changed even as inflation and salaries and everything else are going up. So I think the ability to have funding specifically dedicated to that process is really important. Those are my comments. Thank you.
Thank you, Irva.
Janny?
I just wanted two comments. One is just the fact that dissemination, also if done well, can really enrich taking a deeper dive into some of these environmental health issues with the communities, because you do build a lot of trust. Otherwise, they feel like it's just another outsider trying to tell us things. So that's one thing. And then the other one that I wanted to pick up on Terry's comment about social media. It's not exactly environmental health, but I'm involved in a project looking at the COVID response and the pandemic on some tribal lands, and really doing a mining or deep dive into Facebook. So what was posted as far as public health messaging, and then comments back, and really understanding how the community reacted to those messages, and how, you know, kind of within the community, how the information spread. So I think in terms of environmental health, depending on the community and if that's a tool that they use, there would be some really interesting, you know, either true facts or misinformation, and how that gets interpreted and how it can be, you know, better used as a tool for talking about environmental health things. So we'll see how we're in the midst of doing this analysis. So we'll see.
Yeah, it's my understanding that the SEAL teams for COVID, the Community Engaged Alliance for COVID, has done some nice exploration on misinformation and disinformation on social media. So, yeah.
With that, I would like to call the question and ask for a motion to approve this concept.
So moved.
Thank you, Terry. May I have a second, please?
Second.
Thank you, Trevor. Okay, council members, if you would please vote in the electronic council book. Thank you very much. So our next presentation is a bookend of sorts, coming back to the Alzheimer's and related dementia question. We have invited one of our grantees, who is in fact a long-term NAEHS grantee. I learned this morning that we actually supported a fellowship grant for his doctoral work. But Jason Richardson, who is the Associate Dean of Research in the School of Public Health at Florida International University, and has also been a grantee of our, I think he was in the first cohort of our second cohort, of our outstanding new environmental scientist program. You know, one of our really landmark programs, and also is an awardee of our translational centers, which I think is going to be kind of the central nub of his presentation today. So Jason, welcome to North Carolina, welcome to council, and look forward to your presentation.
Yeah, absolutely. And thank you. It's great to be back in person here at NAEHS. For a long time, this was a home away from home, whether it was grantee meetings or numerous grant reviews for that. So it's always a pleasure to come back to what I like to call home. NAEHS has been very important for my career. Again, as David said, starting through an F-32, and then Cindy and Annette working through me with the fetal basis of adult disease R21 through the ones, and then continued. So I'm very grateful for the continued support of NAEHS, and just happy to share with you a little bit about what's been going on the past few years in our laboratory related to gene-environment interactions in Alzheimer's disease, and how we think this can be a potential path to what I'm going to term precision prevention and treatment. So we hear a lot about precision environmental health. It's a very important concept in precision medicine, but as we're starting to learn more, particularly in the ADRD space, prevention is becoming very, very important. I think this is one of the areas that there's a lot of potential interaction between the environmental health sciences and our clinical partners. So before we get started, this work is highly collaborative. Number of colleagues at a number of institutions. This is our current lab here at Florida International University. But again, our support from NIEHS, NINDS has supported this. The Michael J. Fox Foundation for Parkinson's Disease Research, some of the early parts that transition from Parkinson's into Alzheimer's. I also want to thank NIA. We're in our second year of a 5-year conference series, of what I think is the first of its kind. And it's a partnership with Emory University looking at social, biological, and environmental determinants of health disparities in AD and ADRD. In our first year, unfortunately, we had to push back because of COVID. Last year, we had to have it online, but it was outstanding. And I think we're really starting to reach our goal of de-siloing and bringing groups together and start thinking more broadly about how we work together to move forward in this really critical area. So Dr. Ode gave a really nice background. I'm not going to belabor this at this point, but Alzheimer's is the most common neurodegenerative disease worldwide. It accounts for most cases of dementia. There's many others that were discussed this morning. And we're currently well over 6 million people in the United States alone living with Alzheimer's, more than 50 million worldwide. And this is a huge burden, not only on the patient, but the family, the community, et cetera. It really robs us of those things. In general, and this is what we see with most of our neurodegenerative diseases, we kind of put these into two buckets. We put them into early onset and late onset. And for the most part, our early onset are the ones that are purely genetic in origin. These are our rare particular ones, depending upon which source you're looking at somewhere between 5 to 15% of cases are these more autosomal dominant Alzheimer's cases. But the thing that I always want to make sure of is that people understand is Alzheimer's is not normal aging. This is a pathological process. This isn't just as we all get old, we're all going to get Alzheimer's. There is a pathophysiology associated with this that really results in that decline in memory and cognitive function, losing of executive function. So again, this morning, a little bit about the magnitude of the problem. These are data from Heber. These have been updated a little bit, but just again, projecting. So at this point, let me see if that goes in. There we go. So here we are. This was based on 2020. We're looking at projected number of people age 65 and older in the US population with Alzheimer's disease. And as you can see, as we have this aging population, we're predicted to almost triple this by 2050. This is certainly a very sobering statistic. Besides those things that we talked about, the impact on the patient, the community, and the family, this also puts an enormous strain on our healthcare infrastructure. It also does put quite a bit of impact on financial systems as well. And so sometimes when I'm talking to patient groups, or more particularly with politicians in Washington, D.C., and I tell them, well, if you looked at Alzheimer's and ADRD by gross domestic product, it would be probably about the 17th largest in the world. And there's been economic data to show that. So we're talking about huge, huge impacts as we go along. The second thing that's very, very apparent, or at least becoming much more apparent, is there are significant health disparities in Alzheimer's disease. So if we're looking at this, again, these are data from the Alzheimer's Association. Proportion of people age 65 and older with Alzheimer's disease and other dementias. So in the gray or the black, we're looking at Caucasian. In the purple, we're looking at African American. In the gold, we're looking at Hispanic. So you can see across all of these little epochs of age that we're seeing higher levels both in African Americans and in Latinos. And this seems to be exacerbated by age as we move along. And this is really becoming a very active area of research. There's certainly many reasons for those type of health disparities. Some were talked about this morning and throughout the part with this. One of the things that came out in our conference again last year is even on the things where genetics, you might not normally think about those, even looking at race and ethnicity differences in genetic, and particularly where your ancestral background of that particular risk genes come from, can have a profound effect on your genetic risk for Alzheimer's disease. So when we think about the genetic aspect, there's multiple genetic risk factors. So if we look at this, on the left-hand side, we're looking at risk of Alzheimer's disease. And we'll see these genes that are what we call the causative factors, the dominant negatives. And these are essentially really focused on that amyloid pathway. These are presenilins, amyloid precursor protein, ADAM10 is also involved in amyloid generation with that. As we move more to the right, these are much more common genes, but they're typically more low risk. But important things we're learning from looking at these is typically the field really started looking at amyloid pathway, tau pathway. Now we're seeing a lot more interest as it relates to autophagy, inflammation. A lot of these genes down in that far right-hand corner, that's what they're really involved with. And of course, apolipoprotein E is the best characterized and most well-known genetic risk factor. So in sporadic Alzheimer's disease, if you have one ApoE4 allele, you anywhere from probably 2.5 to 8 times as likely to be diagnosed with Alzheimer's disease. With 2 ApoE4 alleles, 12 to 16-fold increased risk for Alzheimer's disease. And this was a paper that really struck me. It was a commentary in JAMA, and this was after one of the updates on the genome-wide association studies for Alzheimer's back in 2010. And this was the group that has the Swedish twin studies. And the comment was reaching the limits of genome-wide significance in Alzheimer's disease back to the environment. Of course, that kind of piqued my interest as an environmental person. And this was really talking more about some of those lifestyle factors we talked about this morning, but the take-home message was clearly researchers need to pay much more attention to environmental risk and protective factors. And so we started a little bit of a discussion on this back in 2019 with a review article by a really talented postdoctoral fellow in the lab, and Isha just finished her PhD in the lab and is postdocing, is looking at these late onset risk factors. And what do we know so far? We certainly know that age is not causative, but it's the most prominent risk factor for Alzheimer's. There are those genetic factors, again, not the causative ones, but this is ApoE and some of those other things. Sex, we know that females do have a higher risk, particularly earlier onset, and certainly if there's ApoE4 alleles. Family history is very important. This is also often thought to be genetic, but it may be shared environment and some epigenetic components with that as well. Race, ethnicity, we talked about health disparities in some of those data, and certainly traumatic brain injury being that. But for the most part, these are non-modifiable risk factors at this point. If we talk about modifiable risk factors, education is absolutely key and has been shown over and over and over again in multiple studies, and in particular, early childhood to middle school education, at least from the data I've most recently seen, seem to be the strongest. Cardiovascular and metabolic risk factors, so keeping that blood pressure 120 or below, controlling blood glucose, those type of things. Certainly lifestyle, tobacco, alcohol consumption, and what I'm going to talk about today, environmental exposures. And the two that we probably got the best data for at this point are pesticides and air pollution. This was a really nice meta-analysis review article on environmental risk factors for ADRD published in 2021. I'm not sure exactly how this works. I guess it doesn't work like that. Okay, well, I'll just kind of step out here and I can be very loud and not a problem. But if we're taking a look, actually,
You need to be there to go out to the Zoom.
okay, I got to be here to go out to the Zoom. Do we have any idea how this works? The top one. Ah, here we go. Okay, thank you very much. Should have gotten that beforehand. So if we're looking here at increasing risk, and these are the number of studies that were looked at, unfortunately, we've got a lot of really kind of small observations on this, but generally, and there was a new paper that just came out about green space in cognitive function and risk for Alzheimer's. But essentially, on the protective side, if you've got more green space, more social contact, education, more sidewalks, those type of things, those appear to be protective. Now, we certainly need many more studies about that. On the right-hand side, fewer food stores, some other aspects. And then in the exposures, we've typically seen rural living, pesticide exposure, PM 2.5, solvents tend to be a little bit limited and not particularly that strong, although there's been a lot of studies on different solvents and on electromagnetic fields that have been fairly weak. But components of air pollution and pesticide exposure tend to be the two that there's probably the most data on as far as environmental exposures at this point. And this was really how we got into the field. We didn't go out and decide, well, we want to look at Alzheimer's disease and environmental factors, and particularly pesticides. We were several years ago when I was at Rutgers asking some fairly simple questions about Parkinson's disease. And we knew that rural living, consuming well water, and pesticide exposure in general was a risk factor for Parkinson's disease. So this was the time right when the exposome idea was just getting started. So we were still doing very targeted analyses. We had data from several laboratories suggesting that persistent organochlorine insecticides in the laboratory could impact pathways relevant to Parkinson's. And so we asked a really simple question with colleagues from UT Southwestern is, let's take Parkinson's patients, let's take controls, let's measure some of the organochlorines, and this was the 16-panel EPA method for this. And as we were going through this and we started finding some things, we were starting to interpret some of the data and said, well, how do we know that what we're finding is specific or not? So we're just measuring Parkinson's and non-Parkinson's controls. We said, well, let's measure some other neurodegenerative disease and see if it's specific or not. We're fortunate to be able to get a number of Alzheimer's samples for that. And we did actually find what showed up to be a statistically significant effect here that did not show up in Parkinson's. And this is DDE. This is the very persistent metabolite of the pesticide DDT. Now, this was in a small number of samples, probably 20 to 30 at this point. We were fairly skeptical. So we took some more samples from there, engaged our colleagues at Emory. So we had a 2-site confirmation of that. And then we published this in 2014 in JAMA Neurology and basically confirmed what we found. Essentially, those patients that had that were diagnosed with Alzheimer's disease, and we looked at their levels of DDE, they were about 3.5 to 4 times higher in the 80 patients. And this has been confirmed in several other studies around the world, in India, in Canada, also in the United States. And if we look at the right-hand side, if we're looking at the odds ratio of Alzheimer's diagnosis, if you're in that upper the tertile of DDE exposure, this was adjusted for age, sex, race, ethnicity, and location, you're over 4 times as likely to be diagnosed with Alzheimer's disease. And if we added on additional covariates, including ApoE genotype, still highly statistically significant, almost 3.5 times more likely to be diagnosed with Alzheimer's disease. So what are we talking about here? We're talking about DDT. This was the parent compound. It's organochlorine. It's an insecticide. It's highly, highly lipophilic. It has a long half-life. The metabolite DDE, which we're measuring, half-lives are measured in years to decades, depending upon the matrix. It was synthesized in the late 1800s. This was really when organic chemistry was starting to come along, and people were starting to do synthesis with chlorinated compounds. But its insecticidal properties weren't discovered until 1939. And DDT was, this is where we get into some of these double-edged swords when we're talking about things like pesticides, because DDT almost single-handedly eradicated malaria, saved millions of lives in the South Pacific in World War II. So very, very important. Unfortunately, as we're wont to do, when something works really well, we want to use it on everything. And so from 1940 to 1972, in the United States, around 1.2 times 10 to the 9th pounds were applied of DDT. One of the first things that the EPA did when it came into existence was made a decision to ban DDT. And this is kind of a misnomer. Most people think that this just happened like that. It was banned and it was off the market. It was phased out over time. And actually, I can still take you to almost any barn on a farm in the South and find you a 55-gallon drum of DDT with that. So it is still used to control vector-borne diseases in some countries as well. And so we get this question quite a bit. Well, it's banned. Why should we care? And honestly, The levels have gone down and have been going down over time, but you can see around the world, there's still places where there's very, very high levels of this in the environment and also in individuals as well. In the United States, if we look at NAEHS from 1999 to 2002, which is about the last time we have individual level data for this, we have pooled data from CDC. But we see if we look at this, so this is percent of the population here. These are Mexican Americans, non-Hispanic whites, non-Hispanic blacks. And if we look at these levels, it's very easy to see that the non-Hispanic whites have the lowest levels, the lowest number of people with high levels of DDE. Mexican Americans in this population, over 60% of the population there is in that highest level of DDE. And with African Americans, we see it's a little bit less than the Mexican Americans, but it's still much higher as well. So this is not uniformly low in everybody, and we are seeing some disparities with that. And this is not just with DDE. If we look at a number of these persistent organochlorines, so that's DDE, DDT itself, oxychloroquine, heptachloroepoxide, transnolanol, beta-hexachlorocyclohexane. If you look at the different populations, typically very broadly, the Mexican Americans and the African Americans across the board have much higher levels of all of these persistent compounds. And if we work with our epidemiology colleagues and we look at cognitive functioning in this, and this is the digit symbol substitution task, it was only one, that was what was available in this particular subset of NAEHS, we start seeing very different things. If we look across all subjects, we see DDT here, we see heptachloroepoxide. If we look at non-Hispanic whites, really heptachloroepoxide is driving most of this. In non-Hispanic Black, we're seeing different things across the way. And then in Mexican Americans, we're seeing a multitude of these as well. So the take home for this is there are certainly health disparities in exposure and body burden levels. There are certainly relationships between this and cognitive dysfunction as well. So that's enough for the epidemiology. Those of you that know me know I am not an epidemiologist. I'm a toxicologist. I'm very fortunate to collaborate with a number of outstanding epidemiologists. But coming back to my lab-based methods and interest, my question always is do the epidemiological findings have biological relevance? And so if we think back, and this is the way we were thinking when we first started this about Alzheimer's disease pathology, and this is some of the classic pictures that we look at where you have extracellular amyloid plaques here, abnormal accumulation and processing and poor clearance. There's intracellular neurofibrillary tangles. And if we go through this, we're going to see different types of plaques and tangles, and this will become important as we go. So this is a diffuse plaque. So you can see this brown stainings kind of diffuse here as we go. If we look down here at this cord plaque, you can see that brown is very, very tight and in the core. On the right-hand side, this is tau pathology, so neuritic plaques and neurofibrillary tangles. And these are really the hallmarks of Alzheimer's disease. And on the left, on the right-hand side, this is a normal brain. On the left-hand side, this is an Alzheimer's brain. So you can see an overall loss of brain volume and structure in the Alzheimer's brain. So we went back and started thinking, well, we know what the mechanism of action is with this. The dearly departed Toshio Narahashi showed that DDT works by holding open fast action sodium channels. So in the brain, when you're communicating from neuron to neuron, this is a chemical electrical component with this. The sodium channels are responsible for this rapid part of information. And so they open and close very, very rapidly. And you can see this in kind of the standard electrophysiology action potential. So it goes up and then it comes right back down, goes up, comes right back down. Now, if you put DDT on board, what you see initially is it doesn't come down quite as quickly as the first one. Then over time, you start seeing multiple discharges with this. Then over time, this gets more and more till ultimately that neuron won't fire anymore. This is called a depolarizing block. So the way DDT works is it causes hyperexcitability in the nervous system by not allowing these channels to close. So what does that have to do with anything with Alzheimer's disease? Well, at the time, these data were just starting to come out and we didn't know it at the point, but there's fairly good evidence now that early on in Alzheimer's disease, there's what we call network hyperexcitability, where certain areas of the brain are really hyperexcitable. And from a pathology standpoint, this is important because if we look back, these are actually some data from groups in the 1980s, which showed that if you took cultured cells or cultured brain slices and you electrically stimulated those or mechanically stimulated those, or you put on chemicals that stimulated those, you increased something called amyloid precursor protein. And this is what gets cleaved to form those toxic amyloid beta components. And you can increase the amount of amyloid beta that's released. And so that's what happens here. So this is the structure of the amyloid protein. It can get cleaved in many places. Some of these are non-toxic and some of these are toxic. So the really toxic ones that we're concerned about are these A-beta 42, these base cleavage components, and some of the intracellular domain components of that. And they ultimately lead to aggregated amyloid and plaques as they go through. So we start with a very simple hypothesis. Since we knew this depolarization of cells increases APP and A-beta secretion, we asked very simply whether DDT could do this or not. And I'm not going to spend a lot of time on this. We published this recently, Environmental Health Perspectives last year. Just to give you a little bit of a flavor for how we started this out in human neuroblastoma cells, SHSY5Y cells that we differentiated. And we saw essentially a dose-related increase in gene expression for many parts of this pathway. So amyloid precursor protein levels went up, base 1, which cleaves these, went up as well. If we look at the protein level here, so this is red, just as staining for those neurons. Green is that amyloid precursor protein. What we find is if we dose those cells with DDT, we get an increase in amyloid precursor protein. And importantly, this is blocked by the inclusion of tetrodotoxin, which blocks that interaction with the sodium channel. At the same time, in these cells, it also decreases tetrodotoxin, prevents DDT increase in amyloid beta secretion from those cells. We've also confirmed this in primary cortical and hippocampal neurons as well. But moving on to an in vivo component, we use naive mice, C57 Black 6, 3 months of age. We dose these with 3 milligrams per kilogram by oral gavage every 3 days for about a month. And using allometric scaling, this roughly comes out to what people would have been, those most highly exposed in the 1960s would be. We also use a transgenic line to look at this as well. So just quickly going through this, same thing, we measured gene expression of amyloid precursor protein, base 1, some of those other things. And in these animals, we see very similar things that we saw in our cell culture. Let me go back. And so we see that both at the mRNA level, we see that at the protein level as well. Now, in those mice that are an 80 mouse model, we call these the triple transgenic mice. And these were very, a little bit younger mice. We started dosing these at 9 months of age. We stopped dosing them at 12 months of age. And this is really where pathology is starting in these animals. And so if we look here on this part, so these are males, these are females, and we see those that were dosed with DDT have much higher levels of soluble amyloid beta and oligomeric A beta in these animals. And then if we go over on the right-hand side, the left two panels are males controlling DDT. The right-hand panels are female controlling DDT. So what you see is in the control panels, you really don't see any amyloid plaques at all, but in the females exposed to DDT, now you're starting to see amyloid plaques. We see that also in the CA1 region of the hippocampus, and then we see it very prominently in the subiculum. This is an area in the hippocampus where in these models, we see some of the first pathology with that. So this really gave us that indication that DDT exposure is impacting the amyloid pathway at fairly relevant exposure levels for people in the 1950s and the 1960s. Now, one of the other questions that we get is, oh, okay, DDT is banned. And well, DDE is not really neurotoxic. Most people think of DDE as an anti-androgen, an endocrine disruptor. And so why should we care? So, well, that's a good question. We should certainly look at that. And so what you're looking at here is a molecular docking study from the crystal structure of the NAV 1.1 human sodium channel. On the left-hand side, this is DDT showing where it binds in that pocket of NAV 1.1. Well, DDE actually also binds there as well, and it binds actually a little more strongly even than DDT. And when we got back and started looking at this, the reason that people have always thought that DDE wasn't neurotoxic is back when DDT was really being developed as an insecticide, the way they do these studies is they would take a fly and they would either pipette the pesticide on them or feed it to them. And the DDT flies killed over in about 10 or 15 minutes. The DDE flies didn't in 10 or 15 minutes. So they say, okay, DDE is not neurotoxic. We don't have to worry about that. Let's move on with life. So after looking at this binding, we collaborated with our colleagues at Rutgers University, and these are data in human-derived induced pluripotent stem cells, so neurons derived from the iPSCs, and also primary mouse neurons. And so what we can see on the top is these are mouse cultured cortical neurons. Here's DDT, and we look at action potential frequency, and we look at the resting membrane potential. So if you're holding open fast action sodium channels, those action potentials, you're going to have more of them. You're going to decrease your resting potential, which you can see here. So here's DDT on the top, DDE on the bottom, and you can see with time in there, and this is 1 micromolar, then this is 10 micromolar, you're seeing many, many more of these spikes of action potentials. So both of these compounds are doing that. And they're certainly also doing it in human neurons as well. And this is the first time, to our knowledge, these data have actually been shown both in mouse neurons and in human neurons. And this is probably a little bit easier way to see it. So on the left-hand side, here's our mouse with DDT. Here's with DDE. Here's our human DDT. Here's that with DDE. And if we look at our populations of the cells at 2 different types of exposure, we get this really strong bursting phenotype that tells us that these neurons are highly, highly overactive. Now, if we take these same primary cortical neurons, or we take the induced pluripotent stem cells, and we look down here, and we're looking at secretion of A beta 40 and A beta 42, A beta 42 being the neurotoxic version. And then we did the same thing with DDE over here. What we see is a really nice dose response with an increase in A beta release. This is blocked by tetrodotoxin, both in DDT and DDE treated cells. And most importantly, what we found in this is at concentrations as low as 50 nanomolar, we're seeing significantly higher secretion of amyloid beta. And 50 nanomolar is very important because, again, those levels in the environment are going down, but those are levels that we still find in people today, and not even just the ones that are the most highly exposed. So we took this into a slightly different Alzheimer's disease mouse model. This is called the 5XFAD. This has multiple different causative genetic polymorphisms. In this, we use the same dosing paradigm that we did in our previous paper. And these were dosed from 1.5 months of age to 4.5 months of age. Now, in this particular animal model, it's fairly aggressive with its pathology. And you first really start seeing this plaque-related pathology at 4.5 months. You can see amyloid beta going up fairly early in these. But what we find with this is, so if we look at the hippocampus, the A beta 42, now we're starting to see some things that we are used to seeing more in humans and later on in these models, which is soluble levels of A beta 42 go down. If we look at insoluble levels, and so this is guanidine HCl extraction and a formic acid extraction, you can see that both DDT and DDE are increasing the amount of insoluble A beta. And just to bring your attention to the scale here, this is a regular 5XFAD mouse. So they've got about 200 picograms per milligram of protein of A beta 42 that's insoluble. DDT is about 300. DDE is about 1,000. So it's a massive increase in insoluble amyloid beta, which can be seen here when we start looking at those plaques. Now, remember when I showed you humans, we had those diffuse plaques, which is what this looks like in the control of the 5XFAD mouse. And here's our DDT and here's our DDE. You can see much more dense core, particularly in the pseudo-colored, and we performed Gini coefficient calculations, which allowed us to look at the intensity in these. So we're seeing much more dense core plaques in the subiculum and layer 5 of the cortex. And if you look at this particular animal model over time, this is a 4.5-month animal that's treated, but the pathology looks like about a 10 to 11th month old animal that's not treated. And that's backed up if we look at behavior. So this is Y-maze for cognitive function. Certainly that's down, decreased performance in the DDT and the DDE. If we look at plasma neurofilament light, which is a biomarker of general neurodegeneration, that's significantly increased in the serum of both the DDT and the DDE treated animals. And more importantly for us, unbiased stereological counting demonstrates that both DDT and DDE are actually causing neurodegeneration and significantly decreasing the number of neurons, both in the subiculum and layer 5 of the cortex. Again, this is at 4.5 months of age. This type of neurodegeneration in this model doesn't normally occur until about 1 year of age with that. So I want to just finish up a little bit talking about the gene-environment interaction component. And these are some data out of that initial JAMA Neurology paper in 2014 when we were looking at mini mental state exam, which basically said if you were in the third tertile of the DDE exposure against the first tertile, you did worse in mini mental state. If you had an ApoE4 allele, well known, you do much, much worse than that. But if you put them together, it's not synergistic, but they're greater cognitive dysfunction than either one of those alone, suggesting there's a gene-environment interaction between exposure and the ApoE genotype. So how do we go about trying to understand this? So we've done this a couple of ways. I'm going to show you our in vivo data with humanized ApoE targeted replacement mice. We've also done some work in isogenic iPSC neurons as well. So these were Patrick Sullivan's group at Duke developed these several years ago. So they're under the mouse promoter, but they have the human gene in there. We treated male and female ApoE3 and ApoE4 mice with 3 milligrams per kilogram DDT every 3 days between 3 and 8 months of age. And then we've done a number of things with these. Certainly these are still being analyzed. But one of the first things we found when we look at RNA sequencing data is that sex has a major impact on differences in gene expression. And that if you have the ApoE4 allele on board, it causes some of those differences to collapse. So you have to really be careful about how you're looking at those things and how strong of an effect your genetics have. But here's just an example of looking at a male DDT E4 versus E3. These are a lot of the genes that are changed. See a lot of things in biological processes. And when we go up here, these are things we would actually expect knowing how it works. Chemical synaptic transmission, looking at neurotransmitter transport, regulation of neurotransmitter levels with that. So many of the targets that we would expect to see with DDT, but they're exacerbated in the ApoE4 genotype. And if we look at those markers of neurodegeneration, that neurofilament light in the plasma again, at 8 months of age, even here, both the male and the female E4s have higher levels of plasma neurofilament light, even without being treated, suggesting that in these animals, as they age, you're already starting to see neurodegeneration in this model. But on the right-hand side, in the blue is the male, and then this dark one is the female. These were the E4s that were treated with DDT. And so you can see a significant increase in the males, but much, much more profound in the females with the E4 allele. And so we have quite a bit more going on in analyzing data with this. Our colleagues at Emory have run metabolomics on these, so hopefully we'll be able to get a much better handle on what's going on with these animals and why they might be more susceptible. And that kind of comes to the point where I'm going to end in the last couple of slides is well, how do we use this information to translate to humans? So we've heard about this a little bit with some of the concept clearances. There's times we want to translate to patients, there's practice in health decision-making, there's development of methods in the laboratory for new diagnosis, therapy, and prevention. And so from a therapeutic standpoint, we know that there's recently been 2 drugs approved for Alzheimer's. They're fairly controversial at this point. I think most have a cautious take on this, but cautiously optimistic that we've got to get to the point of disease modification, because anything that's currently approved besides those 2 is really to try and treat symptoms. And so when we started thinking about these things in the precision medicine approach, this was from Tom Montine's group, and a really kind of simplified aspect of things where you look on the left-hand side and you've got a causative mutation. Again, we know these are pretty rare. We have genetic risk factors. These are very common, but not very powerful. And then this kind of, we put it in the box, gene-environment interaction. So can we detect these latent pathophysiological process, and then can we tailor particular interventions? I think that's the holy grail for us. We expanded this, and I think we're actually in the fields at a point we need to do an update of this. But the idea is we do have a very heterogeneous disease population. Dr. Ode showed that very nice this morning. We see so much mixed pathology. We see so much comorbidity. We see differences based on race, ethnicity, sex, age, all of those type of things. And we have to take that into account. Genetics is very important, but we also have the shared environment to think about, genetic screening. What about environmental exposures? And that's where we're starting to bring in and think about the expose on. Lifestyle factor is very important. So if we bring this down, now we start thinking about, okay, how do we confirm this in our biomarker screens, whether they're serum or plasma based, PET or MRI based, and can we ultimately modify some of those risks at this point? So the key in my mind is, this is where that prevention part comes in. Who's at risk, when, and what? And can we get to those people early? And then on the back end, there's that personalized therapeutic intervention. So if we understand how these things fit together, well, maybe those A-beta lowering antibodies like the 2 approved are going to work. There's tau things going on. I personally believe it's likely going to be a combination. I haven't talked a lot about inflammation. We work a lot in this space as well, and then other targets. But again, it's likely to be a combination. So how do we work this through same type of framework? And so this was just a kind of a very simplistic one that we put together for a paper in 2018. But if we take it from what we've identified in all of our studies, and we use the DDT and DDE kind of as a case study. So genetic sequencing, we've certainly identified ApoE4 is important for this, but we don't know about other potential genetic modifiers. Well, there's blood and CSF biomarkers. Well, we've shown that in the animals, and we're working with Brenda Plossman and Hong Li Chen in the agricultural health study, and looking at exposure versus these plasma biomarkers as well, that go along with an environmental exposure history. And then, neuroimaging is a little bit harder to do. But in this instance, if you've got somebody, let's say, for example, the ApoE4 and their high DDE, and we kind of know how DDT and DDE are working, well, again, can we identify those people early? Can we remove the DDT or DDE? Because we still don't know right now, is it the initial insult or the continuing presence of these persistent chemicals? And then finally, knowing the mechanism for these things, can we then potentially look at what might be a better therapeutic intervention with that? So just to wrap up, from overall conclusions, hopefully, and again, I think I'm kind of preaching to a choir here, so to say, is that environmental factors do play a role in Alzheimer's disease, whether they're environmental exposures, whether they're where you live, different types of things. But most importantly, this interplay between genetic susceptibility and this big E environment is likely a key mechanism of understanding both risk and trajectory of the disease. And if we can identify the genetic susceptibility and those environmental contributors, can we go to early identification, or will this help us lead to a personalized or precision approach to treatment or prevention? And just to kind of add up, this has been going on for a while. Where are we going now? Dr. Owen showed that this one, we're very fortunate to be collaborating with Barbara Cohen and Dean Jones in a multi-PI looking at the prenatal exposome or the in utero exposome, life course cognition, and prodromal Alzheimer's disease, with that incorporating the exposome, metabolome, the plasma-based biomarkers. We're also working quite a bit in the space of the DDT effects on ApoE and how that affects the pathology. I didn't have time to talk today about ApoE effects on neuroinflammation, mitochondrial function, and autophagy, and how that interacts with environmental exposures. And then finally, working with colleagues and some things coming up to take more of a population genetic approach to identifying gene environment interactions. So thank you for your time and attention. I'm happy to answer any questions you might have.
Thank you very much, Jason. That was a tour de force, I think. Really some really kind of very compelling data there. So this is open for questions. Harry.
So in our discussion about the exposomics and things and talking about biobank samples, as we know, DDT and its metabolites are very environmentally persistent.
Yes.
They keep very well in biobanks too.
Very well.
What would be, if you had unlimited funds, what would you want to do in looking at various cohorts now? Because it seems like how we would normally go about replicating a GWAS, that you could potentially look at many different related species to this in multiple cohorts pretty quickly.
Yeah, I think that's the idea in those cohorts. Can we look broader? And again, that's that idea of kind of taking that population genetic response again. I think one of our biggest problems with our biobank samples. So, and when I'm thinking about biobanks, a lot of the times I'm thinking UK Biobank, which is certainly one of the largest, is we're not very diverse in those. So that's probably 95, 98% Caucasian, Northern European with that. So we don't have that distribution of the genetics that we would really, really want to be able to trim some of these things down. One of the ones we're starting to work with a colleague out of ours with North Texas Health Science Center, Sid O'Brien, who leads a consortium funded by NIA, and Sid has been collecting very large numbers, particularly Mexican American, but also African American in Texas as well. And now there's more. I think there's 8 universities in that particular consortium, the new one. So hopefully we'll be able to get to those type of things and leverage some of those things that NIA is invested in, and now we'll be able to bring some of those environmental components to it, because people are excited about sharing samples with those, and those are things that are already sequenced, and we have other things as well.
But there are a lot of disease cohorts that have very good diversity in them.
Yes.
They may not be like in a public biobank, but
Of course.
they're out there.
Yep.
Rick, you're next in the queue.
Yeah, can everyone hear me okay?
Yes, we can, Rick.
Switch to Zoom. So Jason, that was a terrific talk, really interesting. Just a question related to gene by environment effects.
Yes.
You're clearly looking at some of the genetic variants, sequence variations in the genome. Have you ever seen a case where the gene may be sequence wild type, but it's now altered in its expression or turned off as a consequence of an epigenetic modification?
So let me make sure I'm understanding this correctly. So not the variant itself, but something where we've got an epigenetic change, and now we're seeing a change in expression level of that
Well,
gene.
let's take a simple case. So you could, so say that you would have a genetic allele, an inactivation allele. So you're essentially knocking the gene out.
Yes.
But you can also knock a gene out by changing its epigenetics, turning it off through epigenetic mechanisms. So I'm just curious, how often do you see, say, Alzheimer's genes actually turned off or turned down or somehow altered in their level of expression through epigenetic modifications, not just genetic modifications? I'm just curious if you're looking at this sort of thing. It's a standard question that I ask, people doing this type of work.
Yes, and For example, amyloid precursor protein is very clearly epigenetically regulated, both by DNA methylation, histone modifications with that. That's been reported. Nasser Zawiya's group's reported that with lead. We've got some indication that that happens with DDT as well, because when you're working with compounds that depolarize particularly neurons, a lot of things happen with that epigenetic machinery with those things. Now, as far as silencing, we haven't seen as much of that, but we're not looking at the epigenome or genome-wide level where I think we should be to look at that. One of the areas that we're very interested in, and that was, I didn't talk about it today because that would have taken us down too big of a rabbit hole, but one of the most highly upregulated genes we see in this exposure is actually ApoE itself. And we see differences based on whether it's E3 or E4, how much that is upregulated. And that's something we're very actively looking at this point, because there's a bit of a controversy in the field. So do we want to increase ApoE function, or do we want to decrease ApoE function? And I think it's context and genotype specific for that. There's some beautiful work, out of several labs targeting ApoE with antisense oligonucleotides or cell-specific knockouts with those type of things. But we do see a very powerful upregulation of ApoE following DDT and DDE treatment, particularly in astrocytes, which is the most abundant place. But it's still somewhat controversial. But we see massive upregulation in neurons as well.
Is there an epigenetic signal on the ApoE that is likely to be the cause of that, or is there some other more indirect effect that's causing the upregulation?
Right. That's what we want to find out, Rick. I think our first hypothesis is that it is epigenetic and most likely through histone acetylation, but we've got to be able to show that. So that's actually going to be a big part of our renewal grant that we're putting in.
Okay, sounds good. We'll be looking out for it.
Yeah.
Irva.
Yeah, I just want to shout out that it's very exciting to see this gene environment interaction work happening, because it's that when you, in my experience talking to the geneticists, they're very focused on genetics, gene interactions a lot. But our experience in trying to get funded even to do this in autism, where we've now published on, I don't know, 4 or 5 papers where the gene environment interactions are really strong. Mm-hmm. And when we did the power calculations using that strength, we were criticized tremendously, because they said, no, you can't hypothesize 3-fold or 4-fold increases. It's just going to be, it's like 5% increase. But no, that's not what we see.
You're right. It's the genetic. And that's why when I talk to people, purely environmental causes are going to be very, very rare. Pure genetic causes are pretty rare. Probably much more common than purely environmental causes. But so now we're talking about 80 to 85%. Let's just call it 80%. That's where we've got to work, the gene environment. That's what's going to let us know. But again, it does take that education and that sea change. And I'm very heartened that the Alzheimer's field has been very open. To be honest, I've been very surprised, because many of us in this room fought these wars in the Parkinson's field and still do for those things. And they've swung back and forth. But I think having open-minded people, bringing people in from the outside, along with the traditional geneticists, that's what we're going to have to do to make progress on these devastating diseases. It's got to be people with an open mind and these multifunctional teams and multidisciplinary teams.
Yeah, and I think that the strategy of going for, the genes that have impacts on the processes that are related to the environmental chemicals that you're looking at, that's a really winning strategy.
Yeah, we tried to take it in 2 ways. I mean, certainly the ApoE came out of human data. So that was, I would call that low hanging fruit. And so that's what we were working on. We had the tools, we're working through data. And this was the Victor project that David was talking about that we had with Brenda Plossman at Duke and others. So kind of worked some things around in there. It's not a huge sample. It was a little over 230, but we've got precision medicine array genotyping on those. We've got samples where at that time, again, we weren't doing full exposome, but we were doing organochlorine, and we had some plasma biomarker or serum biomarkers. Now we've got the ability to do more. And we were actually just talking the past week or so how to expand that, because the biomarker field in ADRD is moving so rapidly. I mean, it's almost every week, new data, new meta-analysis, people really working hard on which platform and to validate those. It's actually a very, very exciting time.
Hey, Irva, this is Rick. You'll be encouraged. I was talking to a geneticist you may know. His name is Francis Collins. And he was reminding me the other day about, well, don't forget about those gene by environment effects, Rick. So Francis, yes, I'm pretty aware of that.
Excellent. Thank you, Jason. That was a great
I
presentation.
appreciate it so much.
And so I'd like to, we're running a little bit behind schedule, but I do recognize we need a little bit of a break. So I'd like to take a 10-minute break, and then we'll come back for the council discussion, I think.